Hello, everyone, and thank you for attending today's webinar, Overcoming Challenges in AAV and LV Viral Vector Manufacturing, presented by Fierce Biotech and sponsored by IDT Biologika. I'm Rebecca Kuschmeider, and I'll be moderating this webinar. I'd like to start off by introducing our speaker today. We'll be joined by Matthias Kahl, Director of Process Development for IDT Biologika. You can read his full bio on the left side of your window by selecting the Speakers tab. Just a few notes before we begin. To access additional resources for today's presentation, you can click on the Handouts tab button on the left side of your screen. You can access closed captions from the bottom right corner of the video player. This webinar is being recorded and it will be available to watch on demand within 24 hours. We'd love to hear from you. During the presentation, please submit any questions you have using the Questions and Answers tab on the left side of your screen. We will conclude with a Q&A session, time permitting. All right, now let's go ahead and begin. Matthias, please go ahead.
Thank you so much for the nice introduction and the opportunity to speak about the current challenges in AAV and LV manufacturing, as well as how IDT is addressing and overcoming these challenges with our manufacturing approach to save costs, efforts, and to accelerate the timelines into clinics for our clients. But let's start with a short company presentation of IDT Biologika. IDT Biologika is a mid-sized contract development and manufacturing organization with a 100-year tradition. We are a private company and belong to the Clocky Holding Group. Our headquarters are located in Dessau, Germany. Here we offer the entire value chain of GMP-compliant manufacturing of biopharmaceutical products. Beside our headquarters, we have an ISO-certified manufacturing site in Rockville, Maryland in the US for clinical phase 1 and 2 manufacturing. Our smallest site is located in Magdeburg, Germany, close to our headquarters, where our process development unit is located, with permission of BSL-2 material handling. We are approved by different authorities, for example, FDA, EMA, and ANVISA. We have a strength of around 1,800 people working for IDT. Last year, a turnover of over 300 million euros, and since 1993, we invested over 600 million euros in our pharma park. On this slide, you see a helicopter view of our headquarters in Dessau, Germany. On the left-hand side, the different drug substance buildings are labeled with the corresponding capacities for filling, where we offer common filling formats for early clinical phase up to commercial product manufacturing. The building for the quality control is in the middle of the site, and on the right side, there are further buildings where the pharmaceutical products are packed, stored, and shipped. As you have seen in the design of our pharma park, our business model is to provide our clients with the full service portfolio for the manufacturing of pharmaceutical products. Our mission is to guide our clients after identification of a promising lead candidate through each step beyond commercialization of a product, starting with the process and necessary analytical development through the clinical phases and ending up with the production of pharmaceuticals for the market worldwide. We provide our clients with high expertise in the manufacturing of different biopharmaceuticals, for example, vaccines and gene or immune therapeutics. Therefore, we have established a broad spectrum of technologies in combination with several cell lines for the production of different viral vectors. For numerous viral vectors, we already have different platform approaches established to offer a complete solution for our clients. Today, I will focus on one of these solutions that we have generated for AAV and lentiviral vector manufacturing, among others, to overcome challenges that we observed in the market over the last year. AAVs are one of the smallest known viruses, with an average size of around 18 to 28 nanometers in diameter. They belong to the genus of Dependoparvovirus, which means that they are replication incompetent and need a co-infection with an helper virus in order to replicate. For example, an adenovirus, which is where the name adeno-associated virus comes from. One of the advantages of using AAV as a viral vector in gene therapy is that the virus is relatively widespread in the human population, and due to a highly asymptomatic infection, the therapy derived is quite safe for the patient in terms of unwanted side effects. The other viral vector that we are speaking about today is lentivirus, which is a retrovirus, meaning that the single-stranded RNA is integrated in the genome of the infected cells via retroviral transcription and integration activities. The most known lentivirus might be HIV-1, which has caused AIDS. And most of the lentiviral-based treatments are based on these HIV-1 vectors, which lead to the fact that we have to ensure that no native virus can be generated during the manufacturing due to molecular safety mechanisms. As you can see in the picture, the construct of lentiviral vectors are more complex in comparison to AAVs, and that they are larger with around 100 nanometers in diameter. Both are established vectors in the field of cell and gene therapies yet, but they have own favorite application areas, which will be presented in short on the next slide. Therefore, it is helpful to understand the movement and development on the market situation, which are already well reported for AAV, like in this publication from Kazmir et al. In 2021, showing that there was quite a good movement in the use of AAV for gene therapy since 2003, but the use increased strongly between 2014 and 2015 with a significant higher commercialization of these new therapeutics on the market. The evaluation of the distribution of programs in clinical phases is also interesting, showed here in plot B, indicating that there are many programs with AAV-based treatments still in the early pipeline, and that these new opportunities for disease treatment covering a high potential for new drugs in the future. There's also an observable tendency in the market evaluation that some serotypes, especially AAV2, 5, 8, and 9, are the most favorable for pharma companies, but also that there are more and more specific engineering AAV candidates are coming into the pipeline with a tissue-specific construct design. The manufacturing process that covers these tendencies on the market will be beneficial. On the right side of the slide, you can see some EMA and FDA-approved AAV-based products on the market. One fact stands out immediately is the cost for treatment with several €100,000 per treatment. Furthermore, it is indicated that most of these gene therapies are for very rare diseases, which increases the development cost even more among such as a small number of potential patients for reinvesting. Another evaluation of ongoing clinical trials for AAV was published by Edna Au et al. In 2022. Additionally, plot B is interesting, showing the different approaches where AAV-based treatments are being investigated, covering several indications that are mostly caused by dysfunctional genes like blood disorder or lysosomal storage disorder. In plot C, you can see that those diseases are mainly treated by the replacement of these dysfunctional genes or the addition of supporting genes. Another interesting aspect in the current market situation is that for some indications, several clinical trials are ongoing with a very comparable design and mode of mechanism, as we can see in the table on the right side for hemophilia A treatment. What we can conclude from this table is, in fact, that different serotypes of the viral vector were used, yet the same transgene indicates an equal mode of action for these treatment approaches. There are some indications that are of high interest for biopharma companies, where a competitive situation exists, and a predefined solution for the manufacturing could be beneficial to ensure marketability. In terms of indications, we have a different picture when we look to evaluate the antiviral vector clinical trials, as presented in the publication from Sharon and Kamen from 2017, which is more focused on ex vivo cell therapies and cancer treatment by the CAR T-cell line technologies. Here, there are also two products approved by FDA and EMA with similar high cost per treatment, since in this case, T-cells have to be extracted, manipulated via the lentiviral vector, and re-injected to the patients, leading to high efforts. In the publication of Martinez Molina et al. From 2020, we also see that besides cancer treatment, the therapy of different anemia diseases is also a focus of lentiviral vector-based clinical trials. In conclusion, for both vectors, AAV and LV, we have different treatment approaches, often rare disease indications, and high costs for treatment itself. Among others, these high costs for treatment are also linked to the manufacturing process, which will be considered on the next slide. Starting again with AAV, we found quite a high variety of different manufacturing approaches for AAV, as we can see in this Paul white paper. At the beginning of AAV production, especially the native replication mechanism for AAV via an adenoviral co-transfection was performed for AAV expression. The high productivity led to a shift to the baculovirus expression system, but the disadvantage with the insect expression system was the production of baculovirus before AAV manufacturing and the presence of those viruses in the harvest, which have to be excluded during the downstream processing. The most common strategy for AAV manufacturing today is the combination of an HEK293 producer cell line and a plasmid transfection, overcoming these issues from the past. Due to the fact that HEK293 cells can be cultivated adherent or in suspension, both strategies are applicable in the manufacturing, coming with their own advantages or disadvantages. For example, adherent platforms are mostly cultivated in combination with FBS, which can be avoided by cultivation in suspension. On the other hand, we have the opportunity to concentrate the harvest before lysis in the fixed bed bioreactor, and due to the bed itself, we have a kind of pre-clarification which could improve the downstream process performance. And it depends at least on the product specification which platform should be used for manufacturing. The transfection strategy of AAV generation is shown on the slide. Most common is the triple plasmid transfection. Required gene elements are split into several plasmids, encoding the identified helper genes from the adenovirus for replication, the AAV capsid and replication elements for packaging, and the transgene plasmid itself, where the therapeutic gene is flanked by two ITR sites. The transgene is then packed into the AAV vector. In this case, an E1 expressing producer cell line is necessary to complete the helper genes for AAV generation, which is the case for SF9 insect cells, Percy6 cell line, and also for the HEK293 cell line, which was immortalized by an adenoviral DNA transfection. Advantages of this system are that the modular system allows simple change of the serotype or transgene by exchanging the respective plasmid, and due to the overall low viral gene in the final product, the safety is high. For the gene therapy aspect, we also use the maximum available capacity of the virus for gene delivery. The production of lentiviral vector is basically comparable to AAV manufacturing. We also use commonly used adherent HEK293 producer cell line or in suspension and the plasmid transfection for lentiviral vector generation. The downstream processing is comprised of typical elements like clarification, anion exchange chromatography, tangential flow filtration, and the stereo filtration at the end of the process. Due to the higher complexity of lentiviruses, a transfection system with 4 plasmids have to be used. Regarding the safety aspect of lentiviral vector background, HIV, the highest priority is to avoid the recombination of wild-type viruses in any cases. Therefore, several generations of lentiviral vector plasmid systems have been developed. Currently, we have the 3rd generation of lentiviral vector plasmid systems, including, among others, self-inactivating elements and regulatory elements on different plasmids to avoid spontaneous recombination of the host viruses. Based on the development on the market over the years for AAV and lentiviral vector, the targeted indications for mostly rare diseases, and the different manufacturing approaches via plasmid transfection, I would like to discuss 4 challenges. The extended opportunities for disease treatment for mostly rare diseases with only one treatment leads to really high costs for the patients, or in some countries for the health insurance system. Among others, these high costs are generated due to high costs for the manufacturing, or more precisely, high material costs that are needed for AAV and LV generation. In direct comparison to other classes of biomolecules, for example antibodies or vaccines, we do not have stable expressing cell lines or replicated viruses that reduces the cost in manufacturing. The high cost driver for AAV and lentiviral vector manufacturing are often the plasmids and the transfection reagents that have to be used for each production. On the other hand, I showed on previous slide that for some indications, several constructs are in the pipeline with similar treatment approaches, and a competitive situation consists. Accelerating into market while also reducing efforts needed is in general an important factor, and for the competitive situation, one key element to save market shares. It is also observable that the requirements in terms of verifiable quality attributes are increasing, since AAV and LV are still quite new drugs on the market, and the authorities are still defining new quality standards. So a high knowledge and expertise in the analytical development is very beneficial for new drug development. On the other hand, the tendencies in AAV and LV engineering are leaning towards more tissue-specific and artificial vector design, which should be covered by generic manufacturing concepts to reduce specific efforts in the manufacturing development. Some of these challenges are based in the nature of things and can't be overcome by the manufacturing process itself. But there are some opportunities that are addressable with a platform-based manufacturing approach, which are shown on the next slides from a developer's perspective. First thing is timeline. Timelines can be optimized by a predefined manufacturing approach with established procedures in manufacturing and predefined analytical standard measurements. This leads to a reduction of efforts needed for clinical studies and saves time to get into the market. It is also constructive that we have a high understanding of our platform process, since we ensure that the platform delivers the desired outcome for manufacturing. Process robustness is therefore required. To avoid additional development activities over the clinical phases, it is meaningful to choose an intelligent approach for the platform manufacturing that works from phase 1 up to commercial scale. That's why the scalability of the process was kept into account during our process development. At the end, all these aspects will have an impact on the cost, since we are saving efforts and time into the clinical research. When we take a closer look at how we can accelerate the timelines, we clearly see that most of the time can be saved during process and analytical development. With the platform approach, we deliver a complete solution for manufacturing, meaning that we use established manufacturing strategies with established technologies and a predefined analytical panel. In conclusion, we can set up each project based on the established platform, and only product-specific adaptations have to be done and analytics have to be implemented. Another time-saving aspect are the transfer activities. Especially in the CMO business, many companies tend to offer only DS manufacturing, only analytical capabilities, or only filling capacities separately. In some cases, other CMOs are not able to produce material for later clinical stages, and a transfer to another CMO is required. Besides the time-consuming aspects for the transfer, we can further reduce potential risk and issues that may occur within the transfer by offering the entire value chain for biopharmaceutical manufacturing at one leadership at IDT. For the GMP-compliant manufacturing itself, we have predefined manufacturing schedules, workflows established, materials qualified, and approved multi-use facilities to minimize extra efforts during manufacturing. Many noted aspects I have talked about today are directly linked to costs. With keeping the efforts and timelines as minimal as possible, we are also able to reduce costs for our clients. I also mentioned process robustness to ensure that the desired outcome of our platform is delivered. Therefore, we evaluated different cell line platforms in combination with established systems at different scales for competitive process efficiency. After successful drug substance generation, we also ensure that the product losses are as low as possible while removing process and product-related impurities to meet regulatory requirements. A high process understanding right from the start of a project is another advantage of platform processes. Since identified bottlenecks and stumbling blocks are already removed and a high process reproducibility is given. This is not always the case, especially compared to other product-specific developments, and the process characterization grows over the clinical phases, which may lead to complications between the phases. The scalability of the process is required since lower volumes of the product are often required in the early stages of clinical trials. Therefore, we frequently see that developers tend to use non-controlled and simple manufacturing concepts, which have their limitations in the next phases in terms of scalability, leading to additional development activities between clinical phases. Our platform process uses only scalable technologies and qualified materials for different scales and is also usable for commercial manufacturing. We have a broad technology portfolio established, allowing us to scale each manufacturing batch according to the client demands. The existing knowledge behind our technologies, for example, the respective critical process parameters, also gains us reliability in manufacturing. On the bottom of the slide, you can see an exemplary illustration of our stir tank bioreactor systems that we have established at IDT for different scales, starting with a minimum 250 milliliter stir tank bioreactor system from Sartorius and ending up with 2000 liter PAL Allego system. These broad technologies allows us also to address several upcoming questions when we are moving to commercial manufacturing. For example, when the reusability of materials for the manufacturing have to be investigated for saving additional costs in the manufacturing for the market. On this slide, you can see how one of our manufacturing platforms is designed. For example, here for AAV. We have established solutions for adherent and suspension cultivation for the upstream processing strategies, using shaker flasks or T-flasks for the early cell propagation to produce enough cells until a stir tank bioreactor or a 6-bed bioreactor will be infected. As mentioned, each of these strategies have its own advantages and have to be discussed at the beginning of a new project. After the final cell propagation, the AAV generation will be induced by a plasmid transfection. Here, a dual or triple plasmid transfection system can be used. For the transfection reagent, we rely on the new Vectovir transfection reagent provided by Polyplus, which delivers high and competitive titers. The downstream processing concept was designed generically so that they are applicable for a broad range of AAV constructs and comprise a two-step clarification followed by an affinity interaction chromatography approach and an anion exchange chromatography for full and empty particle separation until a final concentration and diafiltration step into the formulation buffer is performed. Finally, the product is sterile-filtrated before the fill and finish of the product starts. A detailed look into our platform development is given on the slide, where we evaluated, for example, different purification strategies for AAV based on existing publications. Here we compared 3 different approaches for capturing of AAV: affinity interaction chromatography and hydrophobic interaction chromatography in combination with cation exchange chromatography and an only TFF approach. Besides the yields, we also evaluated different aspects for the manufacturing itself, like process time and how many steps have to be performed for each approach to select at least the best options for our manufacturing platform. As you've seen on the previous slide, we choose the affinity interaction chromatography approach in our platform design since we identified superior behavior in comparison to other technologies for several parameters. In addition to our manufacturing process, we also offer the corresponding analytical capabilities for AAV and lentiviral vector evaluation, covering the current required analytical panel for compliant release as titer measurement and identity testing, hosted DNA and protein quantification, up to safety-related testing like sterility. Moreover, we have established next-generation technologies, for example, next-generation sequencing for intensified advantageous virus testing or digital droplet PCR to increase the precision of our assays. Again, I would like to demonstrate one of our assay developments in more detail for the full empty characterization of AAVs. Common assays, such as anion exchange chromatography-based HPLC assays, have a limitation here, since changes in the payload of the AAV and modification of the particle surface would lead to a product-specific assay development, ending up in higher efforts for each project. Analytical ultracentrifugation is another option for full empty characterization, but also has its limitations with sample throughput and might be influenced by the sample matrix background. For this analytical approach, we rely on size exclusion chromatography approaches in combination with classic UV 280 and 260 nanometer detection and a multi-angle light scattering system. This approach allows us to generate data independent on the sample matrix background. The size exclusion chromatography separates smaller molecules from the AAV particles and transfers them into a defined buffer system. The AAV fraction is then analyzed by the UV and MALS analyzer, allowing both the determination of full and empty ratio and also the quantification of genome and capsid titer, which is an additional measurement to the common use assays before. The use of the established platform is quite straightforward, which is illustrated on this slide here. Only the sequence of the transgene and the desired serotype have to be provided by the client to start first feasibility runs with our manufacturing platform. Different HEK29 cell lines can be used directly from the client or directly from IDT for GMP-compliant manufacturing. The plasmid for the manufacturing will be produced by an external supplier. We have also built up different strategic collaborations with tech companies if the product-specific optimization in terms of molecular engineering is desired by the client. After the specific process adaptation was performed and the process is transferred to the GMP site, no platform switch is necessary to produce the material for clinical phases up to commercial manufacturing. But clients with already performed early clinical stages will also benefit from our established platform processes. For example, if former use strategies are reaching their limitations in scalability, or if a more robust manufacturing approach is needed for the commercialization. This is shown on this graph here, that different entry points for these pipelines are still possible. For example, potential clients for early phase material manufacturing could be technology companies or universities with a promising developed candidate. Another possible scenario could be a mid-sized pharma company that has reached their manufacturing capacity limits and are searching for a reliable partner to continue and to support their further project. In summary, we can offer a complete solution for GMP-compliant manufacturing to our customers in the field of AAV and lentiviral vector production. We provide ready-to-use platform approaches with established analytical capacities which are suitable for early clinical phase up to commercial scale manufacturing with maximum of 2,000 liter suspension volume or 500 square meter surface respectively. We provide the entire value chain for manufacturing, including process and assay validation, quality control, and also qualified persons for final drug product release. Last but not least, I want to focus on the demand in terms of product filling and packaging. In this table, some approved gene therapies with a variety of different filling formats on the market are illustrated, showing that different formats and volumes are common. Besides classical glass vial formats, also CZ synthetic vials are favored by some manufacturers, since they have a lower surface absorption effect and are available with higher protection for light-sensitive products. At IDT, different filling lines for clinical and commercial manufacturing are established, starting from small batch size with 500 vials per batch and ending up with our high-speed filling line with up to 500,000 vials per batch. And besides the classical glass vials, we also offer CZ vial filling too. With it, I'm at the end of my presentation. I thank all of the participants for their attention and joining this webinar about IDT's platform-based manufacturing approach and how we use this for overcoming challenges in the market to save time, efforts, and at least costs into the clinics for our clients. And now I'm happy to answer to your questions.
All right, thank you so much. We can move on to the Q&A now. As a reminder, please submit your questions using the Q&A tab to the left of your screen. We have lots of great questions already. We'll try to get to as many as possible. All right, let's begin. The first question, what are typical timelines from project start until material release using your platform approach?
Yeah, thank you for the question. Can you hear me? Hope this is
Yes.
fine. Okay, so in case we are using the complete manufacturing platform with the predefined materials and analytical methods, we can reduce the timeline down to 10 months from product start to material release. Okay.
Great. Moving to the next question. Are there benefits of using IDT HEK293 cell line for the AAV or LV production in comparison to available other cell line?
Yeah, thank you for the question. Yeah, at least our idea is here to provide a complete solution for AAV and LV viral vector manufacturing. And for us, this includes also the providing of a ready-to-use GMP-compliant cell line for the manufacturing. And one advantage is that the master cell bank here is already manufactured, so there are no additional times necessary for the production, at least, and we provide this cell line also with any additional fees to our client, and the saving at least also costs and efforts for our clients. In terms of titer yields or expression yields, we have here also reaching competitive expression yields for ALV and viral regeneration compared to other cell lines on the market.
All right, thank you. Moving on, how do you handle other manufacturing concepts? For example, if only material production for phase 3 is requested, what is the typical workflow?
Yeah, this is at least a typical scenario for IDT since, as you mentioned, we are a contract development and manufacturing organization, so this is quite familiar, and this is often the case that clients request our capacities for later phase material manufacturing. So in general, this is an established workflow, and also based on these learnings, we also decide to invest in these new platform or complete platform technologies. But coming back to the question, typically we start here with a paper-based evaluation of the current process that will be transferred to IDT, and therefore we collect all available informations in one database, so-called tech transfer database, and based on the provided information, we perform here gap analysis and identifying necessary development activities for a successful tech transfer and manufacturing. The additional development activities or potential development activities will be performed under non-GMP conditions in our laboratories, and typically the first step is here to set up a small-scale model and to confirm the data from the current process from our clients, and starting then with these small-scale models, the investigation of or to investigate the further adjustments. And after finalizing of these development activities, we start in the company with the receiving manufacturing side, then the tech transfer.
Wonderful. Our next question. What is the reason for performing feasibility runs at the beginning of a project?
Yeah, the feasibility runs are performed in the beginning of the project, are necessary from our perspective to ensure that the platform process delivers the required outcome. And if it's not the case, to identify necessary adjustments, which could be the case, for example, when we want to produce an artificial or modified vector, or we need to achieve specific requirements, for example, higher concentrations or specific impurity level. And also in terms of analytical development, we ensure here with doing the feasibility testing that all analytical methods work with in the acceptable ranges, or to determine necessary adjustments too here.
All right. In cases where product-specific adjustments have to be developed, what is your strategy to accelerate these additional work packages?
Yeah, based on the findings from the feasibility run, we designed here a specific development plan, for example, concentration factor adjustments or buffer optimization for formalization. And also to saving time and efforts here, we are able to perform these studies here also on a non-GMP condition. And therefore, we have established different systems allowing us to generate a really high number of data within a short time frame, so high throughput technologies at the end, and to increase here also the success of these additional redeveloped packages. And this is also possible in combination with our established fast-track analysis, allowing us to evaluate these data also in a short time frame with these high throughput technologies. And when we speak about fast-track analysis, we are also able to use these technologies or methods to accelerate at the end also the release of our products. For example, for mycoplasma or mycobacteria or advantageous viruses testing, which can be accelerated down to 1 week using this next-gen technologies or FTA analysis in combination.
Great.
Okay.
Our next question. What is the production yield for AAV particles per milliliter of 293 T cells?
Yep. Typically, we do not use the 293 T cell line, so we use our own T cell line, what is known as T modified. But at least it depends, of course, on the serotype and the specific transgene, what titer yields we do have to expect it. But we are generating stable titers in the range of 1E11 to 1E12 particles per mil with our upstream processing.
Okay, great. How do you measure the viral genome integrity of AAV vectors? Is the technology charge detection MS, multiplex, dPCR, HPLC, long-read sequencing, or another?
Yeah, good question. So it is also really project specific. So we have established different technologies here, at least already mentioned here in these questions. But we are moving forward to next generation technologies here, and using also NGS, so next generation sequencing for identity testing, or it could be also implemented by digital droplet PCR, or typically or classic agarose gel electrophoresis is also usable, at least for identity testing. But at least we are open for these analytical technologies behind.
Okay. Next question. Is your NGS validated?
Yes, it was also approved by the FDA. Yeah.
Terrific. Here we go. Does the plasmid ratio have an impact on the full empty ratio? Have you done that comparison?
Yes, of course. We have, during our platform establishment, tested a lot of different process parameters, as well as the plasmid ratio. And of course, we have seen that this has an impact on the full empty ratio. But at least also dependent on the specific transgene and serotype and plasmid system, dual or triple plasmid system at the end.
Okay. For our next question, you mentioned you have different HEK lines available at IDT to use for AAV production that is regulatory compliant. Can you comment on which HEK lines and any other information about the source of these cells?
Yeah, we are open to share additional information about the cell line when it comes to a request. So if you are interested in the use of our cell line, you are welcome to contact us during our over our general email, or I can connect you with our management team. Yeah, we'd be happy to do that.
Next question. You mentioned increasing quality attributes being requested by the FDA and EMA. What are these CQAs and how do you measure them, i.e. what tool do you use?
Yeah, at least it is expanding analytical panel that will be expanded by the FDA, and I'm not sure what kind of specific analytical method you mean here. If you can specify that.
The question just does not elaborate on that. So maybe the person who submitted that can contact you after the webinar. We can move on to the next question. What is the percentage of full particles at the end of your DSP process?
At the end of the process, so it's also really hard to say because this is really serotype specific. At the end, we have seen that some serotypes tend to produce low ratios of full particles. So this is, of course, a little bit challenging during the downstream processing. And other serotypes tend also to produce mostly full particles. And this, of course, have an impact on the performance of the downstream processing. And yeah, it is also depending on the transgene. So if we use the full capacity of our AAVs for the viral vector packaging, we of course see the tendency that mostly partial or empty particles are generated. So it's there's not a general statement on that possible.
All right. The next question. Have you tested different AAV serotypes in your platform process? If so, did they all work equally well?
No, we see it, at least here, as I mentioned, a dependence on the serotype regarding the titer yields and also the performance of packaging, as I already mentioned.
Next question. How many clinical trials for GT have you supplied?
It's also hard to say how many clinical trials we are manufacturing for, since we have different projects ongoing at different stages, especially when we speak about gene therapeutics. So we have also commercialized gene therapeutic IDT, not AAV based, but we are currently onboarding several projects here, which with their own platform and also with the predefined solution here from IDT.
Next question. What are the AAV and LV recoveries after each downstream purification step?
After each downstream purification step. So this is a question that I cannot answer here during this webinar. So this is a little bit complex and also depending on the specific serotype and transgene at the end. But you are welcome to discuss this after the webinar more in detail by direct contacting me.
Great. Next question. What are the next steps for you to make this manufacturing process more efficient and safer?
Yeah, in terms of efficiency, of course, we are trying further reducing the cost by implementing here a material supply strategy to lower the material costs at the end, to optimizing also the material efforts for manufacturing. So I mentioned the most cost aspects here are transfection reagents and plasmids. So there's a option here to minimize the material amounts for transfection reagent, or also for the plasmids, and also for the productivity itself. We can optimize the plasmid itself with different strategic partners with this experience behind.
All right, next question. Apart from AAV and LV, is it possible to produce live attenuated viral vector vaccines?
Yes, of course. This is one of our core expertise at IDT. So we are really famous for the production of live attenuated viruses or viral vectors at the end.
Roughly what percentage of the AAVs that you work with are natural serotypes versus engineered?
Yeah, so we didn't evaluate the performance of our platform process yet with modified AAV serotypes. Just concentrated at the beginning of the most common established serotypes in the market right now.
All right. Do you have plans to move to stable transfection using producer cell lines for AAV manufacturing?
Yeah, this is an idea that we are already talked about, but since we want to use this platform at the beginning for early phase, clinical phase manufacturing, we see here a higher flexibility to stay at the beginning on the transfection or transient transfection strategy. So it could be an option for later phase material manufacturing to save here also additional costs for the patients, at least during the manufacturing.
I'm afraid your sound has cut out. Are you able to hear us, Matthias? We'll just hold a moment here, see if we can get him reconnected.
Sorry, the connection broke down.
Technical difficulties, we understand. Moving on to our next question. Do you provide cell-based validation assay for cell-specific promoters? If so, what cells are used?
Yeah, it's a really open question. So since this is also really specific on the target cell line and the serotype, for example, or the lentiviral vector manufacturing, and what kind of cell is at least targeted for the gene therapy here. So of course, we have the technology in place and the methods in general to establish these specific assays. At least this is a really product-specific assay, and it is not the question what kind of cell line is used for. So we are orientating every time on the need of a specific product, what kind of cell line we have to implement, or but at least it is every time feasible to implement these specific cell-based assays and to validate this for the product release for CTM manufacturing at the end.
All right, well, thank you very much. We are just about at the end of our time here. We have had a lot of great questions. If we didn't get to your question, we will get back to you personally after the webinar. I'd like to thank Fierce Biotech for presenting this webinar, and IDT Biologika for sponsoring the webinar today. A recorded version of this webinar will be available for you to access within 24 hours using the same audience link that was sent to you earlier. Thank you again for joining, and we look forward to seeing you at future events.